Ancient DNA extracted from Neanderthal fossils of Gibraltar for the first time. A new collaborative study, led by the Natural History Museum and the Max Planck Institute for Evolutionary Anthropology, has extracted ancient DNA from the Neanderthal fossils of Gibraltar for the first time. The new study has confirmed the sex of the skulls, and in the case of the fossil discovered in Forbes Quarry, has related it to Neanderthals beyond Gibraltar. The Neanderthal fossils of Gibraltar are among the most prominent finds in paleontology. The fossils are some of the most historic of their kind, having been discovered at Forbes Quarry in 1848 and Devil's Tower in 1926. The authors of the new study used a DNA preparation method that reduces modern contamination prior to sequencing to isolate the Neanderthal DNA component. The results show that it's now possible to analyze DNA in highly contaminated fossils from relatively warm climates, holding out promise for the recovery of comparably ancient DNA from regions such as North Africa, the Middle East, and China. The paper was published in PNAS.
Radiation from atomic testing in Marshall Islands still too high for human habitation. A team of researchers from Columbia University has found that radiation levels from atomic testing in the Marshall Islands are still too high for human habitation. In their paper published in Proceedings of the National Academy of Sciences, the group describes radiation readings of soil samples from 4 of the islands and what they found. Over the years 1946 to 1958, scientists working for the U.S. government carried out 67 nuclear explosion tests in the Bikini and Inuitak atolls in the Marshall Islands. The tests were conducted to learn more about nuclear weapons and their destructiveness. Prior to conducting such tests, officials with the U.S. forcibly removed the atoll residents to other sites in the Marshall Islands. During testing, researchers discovered that fallout was reaching 2 other inhabited atolls, Rongelap and Utrik, so those people were moved as well. After testing ended, officials with the U.S. government met with officials from the Marshall Islands to discuss the possibility of cleaning up the test sites and when the relocated people might return. In this
HIV vaccine nears clinical trial following new findings. A promising vaccine that clears an HIV-like virus from monkeys is closer to human testing after a new, weakened version of the vaccine has been shown to provide similar protection as its original version. A pair of papers published July 17 in Science Translational Medicine described how the vaccine, which uses a form of the common herpes virus cytomegalovirus, or CMV, was live attenuated or weakened, so CMV couldn't spread as easily. The new version still managed to eliminate CIV, the monkey version of HIV, in 59% of vaccinated rhesus macaques. That result is similar to earlier findings involving the vaccine's original, non-attenuated version. The immunity generated by the attenuated vaccine was also long-lasting, as 9 of 12 vaccinated monkeys could still fight off CIV infection 3 years later. These papers are important because they recapitulate the previously reported unique CMV vector efficacy with a genetically modified vector that is highly attenuated and therefore potentially safer for clinical use, said Louis Picker MD, a corresponding author on both papers. In addition, this new work demonstrates most vaccinated rhesus macaques that are protected against CIV can also be protected against a second challenge years after initial vaccination. This is a level of durability that would be very important for a human HIV vaccine.
Researchers discovered genome-wide variations in gene expression between male and female mammals. A team of researchers affiliated with several institutions in the U.S. has discovered genome-wide variations in gene expression between male and female mammals. In their paper, published in the journal Science, the group describes their RNA sequencing studies in several types of mammals and what they found. Physiological differences between mammalian genders are quite often easy to spot. In addition to organs involved in reproduction, there are skeletal and facial hair differences, as well as height differences. Prior research and anecdotal evidence has also suggested there may be some differences in the way the brain works. But what about variations in gene expression? The researchers in this new effort report that very little research has been done in this area, which is a problem. Recent studies have shown that there are many gender-based health issues. Women are more likely to suffer from autoimmune diseases, for example, and men are more likely to develop cardiovascular diseases. The researchers also found evidence that suggested such gender-biased gene expressions came about relatively recently, evolutionarily speaking. They suggest this finding indicates that research
visceral leishmaniasis diagnostic tests. Accurate and timely diagnosis of the tropic disease visceral leishmaniasis (VL) is one of the pillars for reducing VL deaths. Currently available serological tests for diagnosing VL vary widely in their performance and may, as a whole, be inadequate for VL diagnosis, researchers report in PLOS Neglected Tropical Diseases. Visceral leishmaniasis is the most severe form of leishmaniasis and is potentially fatal if not diagnosed and treated in a timely manner. Approximately 96% of the VL cases in South America are reported in Brazil, where a range of serological tests are available for diagnosis. No previous study has comparatively analyzed the performance of these tests. In the new work, Mariana Freire of the Oswaldo Cruz Foundation Brazil and colleagues directly compared the performance of 8 diagnostic kits on a panel of 236 stored blood samples from patients with clinically suspected VL, including 77 HIV-infected patients. The kits tested were 3 enzyme-linked immunosorbent assays, ELISAs, 2 immunofluorescence antibody tests, IFATs, 2 immunochromatographic tests, ITs, and 1 in-house direct agglutination test, DAT-LPC. In this study, the authors demonstrated significant difference in the performances of different serological tests available for VL diagnosis in Brazil. Our findings highlight the need for more stringent criteria for the registration of diagnostic products in Brazil, including the requirement to carry out validation studies before marketing, the researchers say.
Eco-friendly composite catalyst and ultrasound removes pollutants from water. The research team of Dr. Jae-woo Choi and Dr. Kyung Won-young of the Korea Institute of Science and Technologies, KIST, President Byung-Gwon Lee, Water Cycle Research Center, announced that it has developed a wastewater treatment process that uses a common agricultural byproduct to effectively remove pollutants and environmental hormones, which are known to be endocrine disruptors. The sewage and wastewater that are inevitably produced at any industrial worksite often contain large quantities of pollutants and environmental hormones, endocrine disruptors. Because environmental hormones do not break down easily, they can have a significant negative effect on not only the environment but also the human body. To prevent this, a means of removing environmental hormones is required. The performance of the catalyst that is currently being used to process sewage and wastewater drops significantly with time. Because high efficiency is difficult to achieve given the conditions, the biggest disadvantage of the existing process is the high cost involved. Furthermore, the research done thus far has mostly focused on the development of single-substance catalysts and the enhancement of their performance. Little research has been done on the development of eco-friendly nanocomposite catalysts that are